Hi, my name is Nora,
My name is Dudley.
and we're here from Vanderbilt University Medical Center to lead a fun demonstration about why hand washing is so important to prevent the spread of germs. Viruses are one type of germ, and if you've ever had the flu or the cold or chickenpox, you've had a viral infection. So to make someone sick, a virus has to infect that person, and to infect someone, a virus must get inside a person's body, often through their nose or their mouth, where they attach to cells. And when attached, the virus can sneak inside the cell to make more virus particles. In general, the more cells a virus infects, the sicker a person gets. Also, when a virus is able to enter cells and make more viral particles, those particles can go off and infect other people. So one way you can prevent viral infections is to prevent viruses from entering your body and your cells. And that's where hand washing comes in. So what actually happens when we wash our hands? What happens to the virus? Well, the simple answer is that the mechanical action of hand washing dislodges and carries away most germs, including viruses. But soap also plays an important role in killing particular types of viruses. And we have a fun demonstration using homemade virus models to help explain why.
For this demo, you will need aluminum foil, butter, double-sided tape, a cup of sprinkles or pepper, we have pepper, a medium-sized bowl, hand soap, and a spoon, and warm water. Let's start with making our virus models. Viruses come in many different shapes and sizes. All viruses at their core are made of genetic material, either DNA or RNA, encased in a protein shell known as a capsid. But did you know that based on differences in the outermost layer, there are two different types of virus structures? Some viruses have attachment proteins that protrude directly from the capsid. These viruses are referred to as non-enveloped viruses. Other viruses have an additional outer lipid membrane envelope encircling their capsid, and these are called envelope viruses. And for these viruses, attachment proteins protrude from that outer membrane. Now, virus attachment proteins, or known as hook proteins, these are proteins that enable viruses to bind and infect our cells. And both of the viruses that cause the flu and COVID-19 are envelope viruses.
Now let's make our own virus models. First, we'll start by making two balls with aluminum foil, each about 1/2 inch in diameter. These represent the virus's genetic material. Next, cover both balls with double-sided tape. This represents the protein capsid that surrounds and protects the genetic material. Next, we'll use pepper to represent attachment proteins. Roll one of the aluminum balls around in the pepper, and the pepper should stick to the tape. With the second aluminum ball, cover this one in butter. We're using butter to represent the outer lipid membrane, which only envelope viruses have. Then roll this virus model in pepper. The pepper should stick to the butter. Now you have your two virus models. The first model represents the structure of non-enveloped viruses. The aluminum foil ball represents the virus's genetic material, and the tape represents the viral capsid, and the pepper represents the attachment proteins protruding directly from the capsid. The second virus model represents the structure of enveloped viruses. Again, the aluminum foil ball represents the virus's genetic material, and the tape represents the capsid. But in contrast to the other model, we coated this model with butter to represent the lipid membrane of enveloped viruses. For this model, the sprinkles are stuck directly to the butter, mimicking the viral attachment proteins protruding from the lipid membrane.
Now, let's mimic hand washing. To do this, fill a medium-sized bowl with warm water and add some hand soap and mix. Now we're going to drop both virus models in the soapy water and swirl for about 2 minutes. What do you think will happen to each of the virus models as they are being mixed around in the soapy water? Make sure to really coat both virus models with the soapy water as you mix. What happened to the virus models? What is different between the two, and why do you think the soapy water had this effect? I bet you'll have noticed that the butter, which represents the outer lipid membrane of envelope viruses, slowly dissolved in soapy water. But you've also noticed that the sprinkles, or pepper, which represents the attachment proteins, fell off of the models over time. This is because the soap molecules and butter molecules are really similar, enabling them to mix together really well. So the soapy water strips the butter off of the aluminum foil ball, and the sprinkles fall off as well. In contrast, the sprinkles on the non-envelope virus model should have remained stuck to the model, although some might have fallen off over time. This means that using soap during hand washing with warm water and enough time disrupts envelope viruses like the one that causes COVID-19 and makes them non-infectious. Thank you for joining us today. We hope you learned something about virus biology and the importance of washing your hands.